Welcome everyone to this week's edition of the Extracellular Vesicle Club. This week we're going to be talking about what EVs can do in the tumor microenvironment. We often think about EVs as being these great little particles that we can use for drug delivery. We can use them as biomarkers. We think about the positive aspects, but there are certainly also negative aspects in some contexts for EVs, and we're going to hear about one of those today. So Ekihiro Seki, thank you so much for joining us. Ekihiro is at Cedars-Sinai and is going to tell us about a recent publication from his group and some collaborators. So Ekihiro, thank you so much, and please go ahead with your presentation.
Thank you, Ken, for introducing me. And also, I want to thank all the EV club community for giving me this great opportunity to show our recent study. I'm Ekihiro Seki, professor of medicine from Cedars-Sinai Medical Center, Los Angeles. And I have been working on the liver disease research for last 20 years. And then last several years, after I joined Cedars-Sinai Medical Center, I have been collaborating with Dr. Dolores Divizio, who is an expert of EV research at our institution. And we found several interesting findings about hepatocyte-derived EV for the liver metastasis growth. So we are very fortunate to publish our paper recently in the Cell Metabolism online in May and by print in July. So what we found is in fatty liver, fat accumulated hepatocytes produce more EVs. These EVs transfer their cargo from metastatic microRNA to cancer cells. In cancer cells, YAP is activated and then enhance tumor growth, as well as YAP-mediated signaling induce production of the cytokine called CYR61 to enhance or to promote to make different immune microenvironment, tumor promoting as well as immunosuppressing microenvironment to enhance further tumor growth. I want to start about fatty liver, why this is a problem. So in the US, 50 to 60% of adults are obese, and half of them, so almost 25% of adults in the US have non-alcoholic fatty liver disease, which is associated with metabolic syndrome related fatty liver disease. Currently, slightly, our field changed their name, called MASLD, metabolic dysfunction associated steatotic liver diseases. However, to avoid confusion, today I want to use just fatty liver, this term for our in my presentation. So this is a histology of the fatty liver. So hepatocytes contain a lot of fat deposition. Why this is a problem is because not only the fat accumulation, after the progression of this disease will have liver inflammation. And this disease state we call NASH, non-alcoholic steatohepatitis, or recently called MASH, metabolic dysfunction associated steatohepatitis. Sorry. This eventually causes liver fibrosis and cirrhosis. Cirrhosis is an advanced form of fibrosis, and complication is problem. This condition will lead esophageal varices, ascites, liver failure. These are all life-threatening complications. This condition also can be a good soil for the cancer growth. So increase the risk for the hepatocellular carcinoma, and also by unknown mechanism, fatty liver disease is associated with cardiovascular disease like coronary artery disease, as well as heart attack. So here, I want to focus on one more thing. Number 4, fatty liver is also increase the risk of other cancers. For example, cancers from colon, stomach, lung, breast, prostate, and pancreatic. So recently, NCI highlighted high BMI persons are also have risk for the increased colorectal cancer. And the liver is one of the common metastasis site for the colorectal cancer because their anatomy is really special. So GI organ, the colon, pancreas, and also the stomach, their cancer very easy to come to the liver because of portal vein. These organs connect to the liver with the portal vein. This is not with other cancer like breast cancer, prostate cancer. Their organ do not connect to the portal vein. So only for this GI cancer can connect through the connection of the portal vein and spread cancer to the liver. However, other cancer also have potential to cause liver metastasis, but compare the frequency with this GI cancer, liver metastasis rate is much lower. So in this GI cancer, liver metastasis really have problem because 5-year survival for the colorectal cancer is almost recently 50 to the 6% with treatment, pretty much improved. However, once liver metastasis occurs, 5-year survival rate drop to the 13%. So really a serious problem. So we really want to know the mechanism, how to cure liver metastasis. On the other hand, one day, our colleague, Dr. Michael Lewis, who is a clinical pathologist, actually mentioned to me, "Hi, Eki, when I looked at histology of the liver metastasis patient, I often observe fatty liver." So these are not really big cohort, only nearly the 40 patient cohort. However, we found generally the rate of fatty liver in general population is around 25%, but our cohort found nearly 50% of the patient with liver metastasis have fatty liver. So in these patients, we also found these patients are higher BMI. So obesity or metabolic syndrome related fatty liver they have. So we want to know whether this fatty liver is really contribute to the liver metastasis growth. If this something happen, and if fatty liver patient have something different mechanism for the metastasis liver tumor growth, we have to think something different disease management for those patients. So to understand the mechanism, we use animal model. So usually colon cancer through the mesenteric vein, and this cancer put in the portal vein and spread to the liver. So in our model, we injected tumor cells through the spleen, and the cancer cells immediately get into the splenic vein and get in the portal vein, spread to the liver. So this system is not 100% physiological, but I can tell semi-physiological. So for animal study or basic research, what is important is we have to use a highly reproducible model. So orthotopic model, we inject the colon cancer cells to the colon to wait for the natural metastasis. Actually, metastasis rate is not great, so very difficult to understand the mechanism. So by this reason, we chose splenic injection model for creating the liver metastasis. So we first developed fatty liver by the 6 weeks of high-fat diet feeding, then injected colon cancer cells to the spleen, and wait for the 2 weeks to analyze liver metastasis. You can see after the 6 weeks of the high-fat diet feeding, developed fatty liver by the oil red staining, and then more cancer. So we want to move to the understand the mechanism. We focus on EV. So currently, we understand the EV is very important intercellular communication tool to transfer bioactive molecules like protein, DNA, microRNA from one cell to the other cells. And now we know EV is really heterogeneous, like including exomer, exosome, microvesicle, large oncosome, apoptotic body. So here in our study, we did not really separate each EV. We just here is all inclusive. We use EV term for our study. So these, I want to introduce 2 pioneer studies from Dr. Leiden's group, and they several years ago, they reported really important finding for our field. So This paper showed pancreatic cancer produce exosome. This exosome, including the cytokine MIF. MIF stimulated Kupffer cells, and the Kupffer cells produce TGF-beta. And this TGF-beta stimulates stellar cells to produce fibronectin to create pre-metastatic niche in the liver and enhance liver metastasis from pancreatic cancer. And the right side, this paper is more interesting. So EV contains specific integrin. This integrin direct tumor cell to the metastasis organ. For example, pancreatic cancer EV, including integrin alpha V beta 5. This guide tumor to come to the liver. And on the other hand, breast cancer EV contain integrin alpha 6 beta 4, alpha 5, alpha 6 beta 1. These EV guide breast cancer cells to come to the liver. This is really important two papers, and these papers actually stimulated me how to understand the role of EV in the liver metastasis. We focus on hepatocyte-derived EV in our study. So we know fat accumulate hepatocytes to produce more extracellular vesicles. Here, actually, mouse, we measured EV in the mouse serum and human serum. And you can see mouse fatty liver and the patient with fatty liver, they actually produce more EVs. And we want to know what is the effect of these EVs in the cancer cells. We extracted EV from serum as well as culture hepatocytes and put on the tumors. So you can see compared with non-treated cancer cells and control EV from normal EVs extracted from normal mice. And they slightly increase tumor proliferation and invasion. However, you can see EVs from fatty liver mice showed more proliferation and invasion. And also, we specifically isolate EVs from culture hepatocytes, and we create fatty liver in the culture cells by treating palmitate, which is the most abundant free fatty acid in the fatty liver disease. So after treatment or with palmitate, hepatocyte in the culture get fat accumulation. So we isolated EV and put on the cancer, and we found fat accumulated hepatocyte-derived EVs have more potential to proliferate as well as invasion. So this is we confirmed in culture. What happened in the in vivo? So initially we measured 3 molecules related to the EV production: LAB27A, LAB27B, and neutral sphingomyelinase 2, and we found in mouse fatty liver, only LAB27A is significantly increased. So we target this molecule to regulate EV production. So after the establishment of the fatty liver by high-fat diet feeding, we injected LAB27A shRNA using adenovirus. And then after 2 days, we injected tumors. Why we waited for the 2 days? Because during these 2 days, almost all IV injected adenovirus get into the cells and disappear from systemic circulation. So you can see here, we injected GFP labeled adenovirus. All adenovirus, actually, most of adenovirus get into the hepatocytes. And then after the 2 days, so adenovirus no longer in the systemic circulation, and we injected cancer cells. Cancer cells, they don't get GFP. So only hepatocytes successfully knock down LAB27A. So in this model, we measured EV in the serum. So after the high-fat diet feeding, EV levels increased, which was decreased by the after knockdown of the LAB27A in the hepatocytes. And in these mice, tumor growth is increased by the high-fat diet feeding and further decreased by LAB27A. So these experiments indicated fat accumulated hepatocyte-derived EV contribute to the tumor growth. We did additional experiments over expression of the LAB27A in the liver. We confirmed similar phenomenon. So after the LAB27A in overexpression, systemic EV is increased, and then tumor growth is increased. So we also isolated EVs from fat accumulated hepatocyte in culture, and then we injected EV, and we found the tumor is increased, suggesting fat accumulated hepatocyte-derived EV have potential to grow cancer. So next question, what is the mechanism? We focus on the microRNA. So we measure the microRNA from the human serum, mouse serum, and supernatant from the hepatocytes isolated from fatty liver mice. And also we created fat accumulated hepatocyte in culture, and we found 6 microRNAs are commonly upregulated. Among these 6 RNA, these 3 microRNA commonly target LAT-S2, which is a negative regulator of the YAP. So overexpression of increase this microRNA in the EV, and this transfer to the cancer and suppress LAT-S2 expression and increase YAP nuclear translocation and cause tumor growth. This is our hypothesis. So indeed, microRNA 25, 92, or 103 nicely promote YAP nuclear translocation. Here, you can see the right panel. And also, we confirmed role of microRNA in the EVs. So we put EVs on the cancer. You can see the nice YAP nuclear translocation. And we wanted to know this 3 microRNA really important for the cancer growth. We knock down microRNA in the hepatocytes and extract EV. In this EV, these 3 microRNA should be decreased. In this EV treatment, nicely decrease YAP nuclear translocation as well as cancer proliferation, suggesting microRNA in the EV contribute to the tumor growth and the invasion. Then we checked really this YAP upregulated in the culture. Also, we can confirm in the in vivo. We analyze YAP signaling by the RNA sequencing. So YAP-related genes nicely enriched in the tumors with fatty liver in mice fed high-fat diet. And the other cancer-related pathway, EMT, MYC, and TGF-beta angiogenesis, KRAS signaling are all upregulated. So these heat maps showed YAP-related genes. Also, LAP27A knockdown in vivo liver metastasis model showed so high-fat diet feeding increased YAP nuclear translocation in the tumors, which was decreased by the LAP27A knockdown. And also, YAP target gene are all downregulated by the LAP27A knockdown in the specific in the hepatocytes. So these results indicate fatty liver hepatocytes derived extracellular vesicle contribute to the YAP activation in the cancer. So we wanted to know about the role of YAP in the cancer. We knocked down YAP1 in the cancer cells and created liver metastasis. You can see increased tumor progression was decreased by the YAP knockdown. So in here, even in the low-fat diet condition, YAP knockdown decreases cancer. So YAP is not only important in the fatty liver condition, generally important and promote cancer growth. We found fatty liver condition increase M2 macrophage recruitment in the tumors. You can see here a lot of the macrophage recruitment. We use staining for the F480, which is a macrophage marker, and the M2 macrophage marker CD206. And then we found YAP knockdown in cancer cells reduced accumulation of this M2 macrophage, suggesting something factor, YAP-mediated factor secreted from cancer cells contribute to the tumor-associated macrophage recruitment. So we focus on one molecule downstream YAP, CUR61, which is secreted protein. And we found CUR61 is increased fatty liver condition in the tumor, and YAP knockdown significantly decreased. And also, we knocked down CUR61 this protein is encoded by gene CCN1. So increased tumor-associated macrophage recruitment was decreased by knockdown CCN1 in cancer. So cancer produce CCN1 and contribute to the macrophage recruitment. Also, in this condition, we can see the suppression of the tumor growth after the knockdown CCN1 in cancer. So we confirmed knockdown of LAB27A in the hepatocytes. So hepatocyte-derived EV production is decreased in this condition. Tumor-associated macrophage recruitment to the tumor is suppressed. So EV contribute to the macrophage recruitment through the activation of the YAP in cancer cells. So we wanted to analyze more detail about tumor immune microenvironment. We performed single-cell RNA sequencing. So this single-cell RNA sequencing nicely separate 4 different M2 cells. So M2A, B, C, D. M2A is phagocytic M2 macrophage. M2B is this population can produce inflammatory cytokines such as IL-1 beta. M2C is fibrogenic macrophage that produce TGF-beta. M2D is pro-angiogenic. They produce VEGF. And we also found high-fat diet-fed condition in this tumor immune cells also highly express immune checkpoint molecule like PD-L1 and TIM3. We also checked CD8 T cells, and surprisingly, we found CD8 T cells number increase in the fatty liver metastasis region. And these CD8 T cells highly express PD-1 and other immune checkpoint molecules. So although CD8 T cell, this anti-cancer function they have, these CD8 T cells increase in fatty liver condition, they highly express immune checkpoint molecules. So they cannot really kill cancer. So fatty liver condition really recruit CD8 T cells. These cells are exhausted. Then we translate our study to the human study. We use IMC, and we use TME. These contain the 35 samples from liver metastasis patients as well as primary cancer. We stained metal conjugated antibody, maximum we can stain with 40 different antibody, and stain and scan and cellular segmentation, we can analyze each cell protein expression by the single cell level. We successfully clustered immune tumor microenvironment TME cells, immune cells, hepatocytes, tumor cells, and stroma cells. And further, we clustered the immune cells to the T cell, macrophage, B cells, DC, and NK cells. And we found M2 macrophage similar to the mouse model, increase in the fatty liver patient. And these macrophage highly expressed immune checkpoint molecule. Similarly, CD8 T cells number increase fatty liver patient as well as immune check molecule increase. So I mentioned our TME also contained primary cancer. So we compared Ki67 proliferation marker between the normal patient, NAFLD patient, as well as primary cancer and metastatic cancer. And surprisingly, we found in NAFLD patient, also in the primary cancer site, proliferation is also increased, and YAP expression also increased. So from this study, we may be able to suggest something signal from fatty liver actually come to the primary cancer and change something phenotype. So we also checked immune cells in the primary site and found M2 macrophage is increased in the primary cancer in the fatty liver patient, as well as highly expressed immune checkpoint molecule in these macrophages. And although CD8, CD4 T cell number in the primary site did not change, but we found immune checkpoint molecule highly expressed in the fatty liver patient in the primary site. Also, this further suggested fatty liver derived signal come to the primary site and it change something phenotype, especially in the immune phenotype. So after publishing our paper, and NCI also highlighted uniqueness of our study. So our study is mainly focused on the hepatocyte derived extracellular vesicle rather than cancer derived extracellular vesicle to create a pro-metastatic niche. Right side illustration actually showed how fatty liver condition through the EV. These yellow colleagues, these are EV, actually take care of the cancer, and the immune cells outside the window actually cannot attack the cancer cells. So this is a summary slide. So fatty liver promotes colorectal cancer liver metastatic growth through production of the EV that contain the pro-metastatic microRNA. And the fatty liver derived EV modulate YAP activity and the tumor immune microenvironment in the liver. So because of they highly express immune checkpoint molecule, especially PD-1, so maybe in the fatty liver condition, they have something resistant to the immune checkpoint therapy. We are currently working on this. And we found IMC study, human study, showed primary sites already have something phenotypic changes in the fatty liver patient. So fatty liver actually produce something signaling. And this signal or cytokine or EV, here we want to know whether this is EV is important. So come to the primary site and change phenotype to make more tumor promoting phenotype. And our high-fat diet feeding model do not induce significant inflammation, just fat accumulation. So we can tell early fatty liver stage also can create pro-metastatic liver microenvironment. So we should not wait until fatty liver disease is developed, even in the early stage. Just fat accumulated hepatocytes can make pro-metastatic liver microenvironment. So we have to and we found different disease mechanism in the colorectal cancer, liver metastasis, with and without fatty liver. This further suggests there is something different. They need different disease management for the colorectal cancer liver metastasis patient with and without fatty liver. So after or almost in parallel, at the same time, we published paper. One paper actually published in Nature from the Dr. Leiden group. They, in contrast to our study, they showed cancer-derived EV also affected the liver to make fatty liver, also make liver dysfunction. So our paper and their paper is, I believe, this is very nice complementation. So we could not really study about cancer cell-derived EV, and Dr. Leiden team nicely showed cancer-derived EV also have potential to affect the liver, especially fatty liver. So this is just one slide. I showed their data. So melanoma-derived EV actually cause fatty liver. This green signal is fat accumulation, and using patient sample and patient-derived EV also the cause fatty liver. And also these cancer-derived EV cause liver dysfunction by decreasing CYP2, several CYP2 for cytochrome P450 molecular expression. So finally, last slide, I want to really acknowledge our lab member, Zijun Wang. Dr. Zijun Wang is the first author of the distal metabolism paper, currently back to the China. And Yumi Yang is firstly established liver metastasis model in my lab and currently preparing another paper about the mechanism of fatty liver and liver metastasis. And Dr. Dolores Divijo helped our laboratory a lot, and we finally we generated this paper. And we have the NCI PO1 program to study the liver metastasis. Dr. Sherry Liu and Steve Pando and Neil Bomick all contribute to this study. And thank you for your attention. I want to stop here.
Thanks so much, Ekihiro, for sharing the data and your conclusions. And I would just like to ask everybody to please remember to put your comments and your questions in the chat box. And I am presently allowing unmuting. So when I call on you, please be ready to ask your question. And we hope to have some conversation here. So let's start with John Bissler. John, please go ahead.
Thank you. That was a really, really good talk. I have a sort of a tangential question, but have you started to look at how the EVs get to where they're going? How do they interact with the immune cells? What's the protein-protein or protein-ligand interface?
So EV, I believe, directly have effect to the immune cells. But here, in our study, we focus on the cytokine produced from cancer cells. So EV first change phenotype in the cancer cell. And here, we actually analyze YAP activation. And activated YAP produce CUR61. This cytokine actually change immune microenvironment. However, you also know, you mentioned about how EV directly change immune cells. This is actually really good question. And we only analyze microRNA, 3 microRNA, but other microRNA also very important. So for example, we analyze 3 microRNA, MIR25, 92, or 103 on cancer cell, changing YAP activity. But of course, these have potential to change function by activating YAP in the immune cells, also to change the immune microenvironment.
Thank you. What I was wondering too is, are there specific targeting proteins? I mean, how does the immune cell know to take up that EV? It's not really what you studied. The whole thing was excellent. I was just curious.
So we will work on that later. So thank you.
Great, good question. And I think related to that, I was intrigued by the microRNA portion of the work as well. And if you had to guess, do you think that it's the microRNAs that are having most of the effect, or do you think that there are also perhaps other mechanisms?
Yeah. So because microRNA has so many targets, and and also target molecule have regulated by so many microRNA, probably I still believe microRNA is not really specific. So mostly Dr. Leiden's work is targeting protein. So I think protein, to target protein, we can get more clear results. However, thinking about analysis, to analyze compared with protein analysis, microRNA analysis is much easier. So we started from microRNA analysis. However, we are really interested in what protein in the EV regulate this fatty liver enhancement of the liver metastasis. I think there is a lot of the mechanism. So not only this single mechanism. So still this is understudied field. So especially in human study, and colorectal cancer liver metastasis seems like this is true. However, in other cancer, it's still mixed results, fatty liver and cancer progression.
Yeah, very good. And I like what you say there. We know that microRNAs can be involved, but it's sometimes difficult to nail down the exact transcripts that they're targeting, or even if they might be exerting non-canonical effects. So that's also a possibility that there's there's another mode of action of the microRNAs that could still be sequence specific, indeed, but different from what we classically think of with the microRNAs.
Yeah.
Have you done any work with extracting EVs directly from tissue?
Tissue? No, that is possible. So I actually I have read several papers about that, but still, I haven't tried because I thought, from the media or serum, so from liquid easy to extract, but how to really separate EV from intracellular vesicle and to differentiate extracellular.
Yeah,
This
it's a challenge. It's a challenge, certainly. And I mean,
but
I know I was a big skeptic about it. I actually went to Andrew Hill's lab for a few weeks to see their brain EV purification method. Of course, every tissue is different. But I left being convinced that there was some opportunity to work with tissue EVs, and indeed there's a growing interest in that, because of course, as you know, the site of action of the EV is in the tissue.
Right.
So that might also be something that that could be a future direction. Is actually attempting to look at the EVs directly there in the tissue, and that could be in the form of. Sorry, are you still there?
Sorry. Yeah.
Are you still there? Oh, I'm sorry. I my screen just went away. It could be in the form of trying to extract EVs from the tissue, or it could be simply, maybe doing some advanced microscopy in the tissue. So lots of opportunities now, I think, that are opening up.
Yeah, that's really interesting.
Yeah.
So,
Lucia Languino.
Hi, Ken.
Hello.
Oh, hi, Dr. Seki. Thank
Hi.
you for the lovely talk. Appreciate it. So the EV that you're characterizing, I may have missed, I apologize, I was a few minutes late. Did you describe what EVs we're referring to? What size, what subtypes?
So actually, we did not really separate this, EV. So we all put together, but mainly we believe we analyze small extracellular vesicle like exosome. So I understand EV is really heterogeneous, and however, our separation system is, we really cannot tell what specific, even, initially we started to use exosome, that term, but when we decide to complete the paper, we decided not using exosome, that term, or change to the extracellular vesicles. So
I understand.
So not really specific, but your question really, and I talked with Dolores a lot about whether we can do more, EV heterogeneity in our study. Yeah. Thank you so much.
Yeah. And then the second question, can you revert the phenotype that you observe? Is there a way to make a fatty liver non-fatty? Did you again say it and I missed it?
So what question? So after
can you
fatty
change
liver
it
and to reverse fatty liver and also continue the phenotype or not? You mean? That's probably really, really interesting because
I know it's gonna be difficult, yeah.
Yeah, but however, that is really important because once we see the patient, probably we have to advise them, because still we don't have approved anti-fatty liver drug. So we can advise to the patient, reduce body weight, change diet, exercise, like that. And we expect to reduce fatty liver. And we expect reduce fatty liver, also the reduce the risk for the liver metastasis or reduce the risk for the cancer aggressiveness. However, we did not test. So actually, we expect, but this is not always true. So your question is really, really fantastic. So
it's a long project. Okay, thank you.
So Eki, I think all this exercise and diet stuff, that's just too difficult. Can we just block the EVs?
Yes, that would be the best.
So yeah. Our approach currently is, we use LAB27A knockdown. So I'm not sure there is a specific inhibitor for the LAB27A. I know that there is an inhibitor for the ceramide and then block EV production.
Yeah, I mean, a mutant, RAB27, maybe Ken knows more than me, maybe a mutant. I'm not sure if there is an inhibitor. Thank you.
Thank you.
Thanks so much, Lucia.
Thank you.
Fernanda Kugaratsky, please go ahead.
Thank you so much, Dr. Seki, for the lovely talk. That was very interesting. So I was wondering if you have looked at the vasculature in the liver, considering that the CYR61 is a pro-angiogenic protein. Do you think the macrophage recruitment is a result of a secondary effect of an increased vasculature, or is a direct signaling mechanism of CYR61 on macrophages?
That is really important question. So currently, I did not check angiogenesis directly here, but our previous study actually definitely proved macrophage produce VEGF, and this actually enhance angiogenesis in a tumor when mice fed high-fat diet. So I did not check CUR61 knockdown or YAP knockdown, but we will do. And I think your suggestion, CUR61 also, mostly binds to the integrin, and the integrin should be expressed a lot in the endothelial cell. So that could be so.
Thank you.
So it looks like we've come to the end of the questions in the chat box. So if anybody else has a question, please put it in now. But I know that Dolores was called away to another meeting today, but I imagine that she has probably mentioned large oncosomes to you once or twice. And you are working with mostly the smaller EV populations here, but you think there's a role for these larger packages as well?
Yeah. So one excuse from my research is we focus on the hepatocyte-derived EV. I believe oncosome, so from their term, that is produced from cancer cells. But if divided,
so maybe we're talking about the other direction this time.
Yeah. But thinking about if oncosome is determined by the size, definitely this is a possible oncosome, but produced from normal cells. If we can define like that, we also definitely that we have to look up about that.
Yeah, indeed. I think there's just an open question for a lot of cell types, under what circumstances they might produce these much larger particles as well. So so good. I think we've identified a few things here today that might be exciting research directions for someone in the future. And certainly we've gained a lot of insights from your work here.
Thank you.
So well, I guess that brings us then to the end of this EV Club. And thank you so much, Ekihiro, for joining us and for sharing your expertise. And thank you to the audience as well for your participation and your contributions. So I hope everybody has a great rest of the week. And I look forward to seeing you again soon at an EV Club coming up. Thank you.
Thank you, Ken, and everybody for listening to my presentation.
Thank you.